I'd like to thank the organizers, Chris, Virgil, and others for inviting me to give a talk here. I always enjoy coming to Cold Spring Harbor, and I actually taught a class here a couple years ago, and it was really great interaction with some of the students and postdocs. Anyway, so so today what I decided to do is in the first half I'll sort of lay down some of the rationales and the technology that we use to do the kind of in vivo circuitry dissection relevant to addiction, and in the second half I'll describe some of the newest experiment we've done in this regard, as well as some of the sort of upcoming technologies that we're thinking about to potentially incorporate into the study. So a question that really sort of at least ponders us a lot is the following. Okay, I think a lot of effort being spent in the last couple of decades looking for quote unquote causal genes in a either physiological or pathophysiological context. So it could be like a causal disease genes or high, sort of a high penetrant risk allele, or a gene that's just really important for, let's just say, some kind of a physiological process in vivo. So you have a causal gene, right? And then the challenge here is how to go from there to actually understand what it does in the brain. How it can, over time, selectively affect different circuits or different cell types that cause eventually, pathological behavior, or in the case of neurodegenerative disorder, neuronal cell loss. So these are the kind of questions really challenging, the whole field. And so in our lab, we study actually both type of disorders, okay, both the sort of addiction-related behavior as well as neurodegenerative disorders such as Huntington disease. So what we've been trying to do is actually try to come up with ways where we take the gene, which obviously will convert into a molecule, it could be RNA or protein, and really try to figure out where in the brain, what cell type or cell types these molecule targets in the brain, and how to have a system where you can see those kind of interaction in vivo in multiple cell types over time, okay? And so the idea is that you have to put these cells into the cellular context and look for both cell autonomous and non-cell autonomous effects. And so that's going to be the theme of the talk, is how to kind of convert a quote-unquote causal disease gene into understanding at the cellular circuitry level and hopefully down to a molecular level. So let me explain this question, in the context of this, this course. So we've been interested in, opiate addiction, opiate drugs addiction. I mean, I think Brigitte, I'm sure, gave a wonderful lecture on the topic. So this is, you probably know more than what this slide shows. Just to just recap, this is actually, one of the oldest drug ever used by human. And it obviously has a huge behavioral effect. Some of them, physiological or therapeutic. This is, the effect on analgesia. But then they have all these, pathophysiological effects, such as the addiction aspect, the drug seeking and taking, the dependence and withdrawal, and also relapse. Okay, and actually the opiate drug use as a sort of a prescription painkiller addiction is on the rise. In the U.S., actually, there's estimated 1.9 million people who are addicted or dependent on the drug, and there's also a major health sort of threat to our society. So each year, estimated there are actually overdose cause respiratory depression, cell death, and death, human death, over 12,000, and as well spread with other disease due to the way the drug is administered. So on the molecular biology side, I think the course organizers, Chris and Brigitte, involved in cloning the major opiate receptors. There are 3 of them. I'm sure you know about that: mu, delta, and kappa. And for the purpose of the course, I'll just for my lecture, I'll just mention they're GI-GO coupled to inhibit cyclase and also lowering membrane potential and cell autonomously suppress neuronal activities. So really a landmark study, actually by Brigitte's lab in 1996, basically showed among all the 3 receptor knockout mice, mu opiate receptors turned out to be the most important in terms of mediating the major effect of the opiate agonists in animals, which including analgesia, reward, and withdrawal. Okay, so this actually, the reward effect originally measured by CBP, but then other people showed it also applies to self-administration. However, otherwise, the mice at least looks okay. They may have some other sort of psychiatry-related behavior deficits, but overall the mice, at least is working, not the talking mice. So that's important because I offered a sort of a baseline substrate for us to start understand where could be the mu opioid receptor acting to elicit some of these really important behavioral effect of opiates. But then when you look at where mu is expressed in the brain, this is a very old slide review, Mansour et al. Really kind of give us the complexity. Mu is expressed not in every cells in the brain, but they're in a lot of the cells. Okay, in many different regions at different levels. And one thing I'm sure Bridget mentioned is that mu opioid receptors expressed, throughout the cell. Okay, on the membrane structures. So it could be on the plasma membrane, near the synapse, at the dendrites. It could be at the presynaptic terminals. So even let's just say you look at the one region of any given region, the mu could be expressed in the cell by the cells located there, but also could be transported elsewhere and to that region. So this is sort of the complexity of the potential understanding where the mu expression is essential in sort of initiating a lot of these behavior we mentioned. So traditionally, the way people try to locate the sites for brain sites for mu action, opiate agonist action in vivo is by either injecting agonists or antagonists into different brain regions in rats, I think mostly, and then assay for behavior effects. So this is a really, really nice review by Bridget's group, showing summarizing some of the experiments. So for example, you guys probably know that injecting mu agonists into the VTA is a animal self-administer, mu agonist to VTA. They will do the same thing with in some other regions such as nuclear accumbens or septum, and there are other regions where there's a adversity effect of a mu antagonist. Okay, so sort of, these are informative. Okay, tell us that maybe there are different regions that are involved in opioid reward. As a matter of fact, there are evidence, I think stronger evidence, supporting there are some reward mechanisms dopamine dependent, others are dopamine independent. However, the sort of the challenge still remain, right? So let's just say, one of the big theory is that, okay, the mu receptor expressed in the VTA interneurons, okay, which, inhibits the dopamine neuron. Interestingly, dopamine neuron themselves do not express mu. So the idea is that these interneuron has a potent inhibition, and then mu can then disinhibit by, inhibiting the interneuron. So there's a good electrophysiological evidence from, old study by North and Johnson et al. But let's zoom in onto the VTA and SNC. This is SNC. This is the interneuron, local interneurons, and then there's other neuronal population that project synapse onto the VTA interneuron. VTA dopamine neurons, such as the neurons in rosomedial tegmentum. Today we'll talk about the neurons in the striatal-direct pathway. So there are all these other populations that project to these neurons, also has a presynaptic mu. Okay, so there are multiple populations of presynaptic mu that could influence in dopamine neuron action. And I think electrophysiologically there are reasonable good evidence for some of them, but not others. But at least sort of by synaptic connectivity, they're plausible candidate for this kind of regulation. So this just to illustrate, if you inject mu agonists and antagonists into that fine region, there's still multiple possibility that could explain the rewarding effect. Okay? So to resolve problem like this, like this kind of problem occur over and over in many of the disease that you think about. For example, the other question that we actually focus a lot of study is in Huntington disease, right? A protein, even probably more intriguing than mu because the size and the complexity of its interacting partners, and it is expressed everywhere, but the degeneration over a long time and is quite selective. So the same kind of question need to be addressed, right? Where this protein act over time to cause that disease. So the kind of strategy we do in our lab is what I call this two-step reductionist approach. okay, to dissect this kind of disease gene pathogenesis. So the idea here is if you have a really just a highly causal or some really gene that's really interesting to you that has already known to have a major effect in vivo, what you want is to build conditional models based on this what I call disease-related gene, okay? And then the first step is really try to identify as refined as you can the cell type within a defined circuits that targeted by the disease gene to elicit critical aspect of the phenotype that you care about, okay? And then the next thing is that if you identify a cell type in which the disease genes expressed has a major effect, you can define that either cell autonomous, non-cell autonomous in the disease, then you can look for the molecular mechanism within that cell that corresponds to the defined cellular role in the disease, okay? So this is sort of the idea. It's not just, you take a gene and just study in some cell line. You want this to put into an in vivo sort of a context. So this is the outline of my talk. What I want to do is I will tell you some of the genetic tools you need to build disease models and dissect disease-related genes in defined neuronal populations. And then I'll give you a very specific example in the second half of the lecture on genetic interrogation of the striatal direct pathway neuronal population opiate drug reward. And also in the end, Chris told me I should discuss some strategies that we're thinking and maybe future strategies, and that'll be a good spot for real discussion. By the way, I really encourage you guys to ask questions. It's not my mandate.
I have been encouraged.
Oh, okay, it's not a monologue. Anyway, so in terms of genetic tools, as Chris mentioned, I was really fortunate to be in Nat Heinz's lab, and Peter Modell and I, we sort of co-developed this technology, engineering bacterial artificial chromosomes generate back transgenic mice. So many of you are probably familiar with this, just in case some of you don't. The advantage of the back compared to conventional transgenic mice, conventional transgenic mice, you can only use about 20 kb per motor. The constraint really was because that was the most common sort of cloning vector, like a phage or whatever small plasmids. But then with the bacterial artificial chromosome, it works as easy as the plasmid grows in bacteria. You can get tons of DNA. But the nice thing is the average size is about 200 kb, 180, 200 kb. Okay? So for any given gene of interest, the unit for all the exons, introns covers about 25 to 50 kb, by and large majority of the genes. And you have to know that all these critical regulatory elements for proper gene expression in vivo, okay? They're basically decorated around the gene and maybe within the introns. So you really need, this is actually, according to many have been done in Nat's group, you really need about 50 kb genomic DM5', entire gene locus, and hopefully 50 kb downstream to give you high confidence reproducible gene expression that's similar to gene X in vivo, endogenously, okay? So the nice thing about BAC is they have the right genomic size to, by and large, for majority genes, confer accurate endogenous-like gene expression. So don't be scared. I won't go into detail about the procedure, but suffice to say that the process to engineer bacterial artificial chromosomes is quite straightforward now, okay? Basically, we have this conditional plasmid expressing, you just need to clone in a small so-called recombination cassette. In the middle could be any modification you want to introduce into the gene on the back, and each arm could be just a few hundred base pairs. And in these two well-defined steps of homolog recombination in bacteria, you just need to transfer bacteria from plate to plate. And within about a week or so, you can actually get a precisely engineered back. The efficiency can be quite high if you are familiar with the procedure. So for example, this is sort of I usually show to my undergrad students. This is sort of the process. You can the back actually already exists. You can just order them. The back containing your gene is all in the UCLA in the database. Okay, you can literally just go to the database in half an hour. You can get the back, cover your gene from many species. Okay, not just mouse. It's important to remember there are also human backs. As a matter of fact, for modeling human disease, we prefer to use human back. Okay. So this, it's reasonably quick. And then this is a very, very critical step, is to prepare intact back DNA. These are very large DNA. They're really easy to break. And, but there are, there are sort of methods that we came up with now, as a field, and then we can really prepare high quality DNA, and that is a key to actually get a high, accurate sort of transgenic mice. And of course, the injection step, the pronuclear injection step, is the same as conventional transgenic mice. So you don't really need to do anything quite different from the, in your transgenic core. So I meant to put this on to encourage those of you who have not made any back transgenic mice to consider adopt this as part of your, toolbox, because it's really useful and it's not that scary. We at UCLA, we've taught people from electrophysiology lab to make these kind of mice. So. Okay, so this is actually meant to, sort of tell you, but also remind me how we get there. This is actually the gene that I studied when I was in Nat Hein's lab as a graduate student. And sort of we first tried to look at the expression of this gene using conventional promoter, transgenic mice. And I actually used 10 kb promoter. That's quite long back then. And lo and behold, after more than a year of work, I got all these beautiful lines expressing like Z in places other than the right place. Okay, this is the most interesting line expressing half of the cerebellar cells. It should be the whole cerebellum. So anyway, that could be a sign. So basically, Nat and I didn't give up, and we decided actually to pursue the back transgenic technology and look at first engineered back transgenic mice. Just looked really beautiful. As a matter of fact, the second back transgenic mice that we made really sort of illustrate a lot of the main point what the technology could do. I mean, not only label accurate cell, this is a combined back GIP, not only label accurately the expression of the gene throughout the development, but also in populations such as Purkinje cell, it beautifully labeled the dendrites as well as the axon. So you can look at the morphology and connectivity. So of course, everyone knows GENSAT. Nat went on to do this really amazing sort of a large transgenic effort for the field. And I think they have, I think last time I read, it's like 10,000 lines generated. And they really, what's important here is it not only give you the expression pattern of the genes, some of these unexpected connectivity that people didn't appreciate before, but also it's a reproducible genetic access to define cell type. That's what you need to remember. If you see a GENSAT mice has a pattern that's reproducibly interesting, you can actually take that back and make new transgenic mice to deliver the protein that you're interested into those cells. I think that's what was important to know. You don't just have to wait for GENSAT to make the next mouse. And then also, obviously label the process, look at the connectivity and in vivo physiology. And you actually can also get all the back constructed mice from these repositories. So what I'm going to talk about next is, we sort of took the back approach. In my lab, we actually add one more sort of a level of flexibility to it. That's conditional back transgenesis. Yes?
Just a question. Can you use this method in rats yet?
Of course, it's been used. It's been used in rats. Yeah, it's been used in other species too. Yeah. It's basically whatever species that conventional pronuclear injection can be achieved. No, as a matter of fact, in Drosophila, they actually can use sort of a integrase type of strategy to also make back transgenic. So now the back transgenic is actually, in fashion in the Drosophila field as well. Yeah. So what we do is we want to introduce conditionality so that you can make a back model with interesting phenotype, and then you can kind of switch off. Right now we're doing switching off, but also you can do switch on in different cell type, okay? So this is really useful, as I mentioned to you, for the other part of my lab, which is studying neurodegeneration. So the neurodegenerative disease, almost all disease proteins are broadly expressed in like every cells in the body, okay? But why certain cells are selectively affected, right? So I won't give you all the gory details because it's not that area of a lecture. But what I can show you is that there are two examples just convince you this kind of a strategy work. Why I want to show you this? Because imagine the back transgene when they integrate it, just like your conventional transgene integration, you still get multiple copies. Okay, it's not hundreds of copies, but it's between 1 and 10, I just say. So if you have 10 copies of back, you have a LoxP flanking the exon 1, like Brigitte do with a conditional knockout, you have 10 copies to worry about. Okay, so what I will show you is that despite the multiple copy integration, the way we design LoxP sites are very close to each other, usually flanking one critical exon. So the efficiency for pre-immediate deletion is really high. Okay, so this is a back transgenic model made by a former student of mine, Tara Weiss. And this is a synuclein back transgenic human synuclein back transgenic. This is actually a mouse model for synuclein multiplication in Parkinson's disease patients. So this is wild type, and again, what we did is we floxed ATG exon with 2 LoxP sites. We strategically positioned the LoxP sites so that it's in the region that's in the intron and it's not very conserved region, so that it should not interfere with the expression of the gene. We know that that's the case. So if you use a human selective antibody, you can see it's really broad expression of human alpha-synuclein in the transgenic mice. But if you take that mice and cross with Cree line, for example, you can cross with nesting Cree, which is completely deleted in all the cells in the brain. Okay? If you cross with a Cree that's cortex specific, the EMX1 Cree, you can see really nice deletion in the cortex and in the hippocampus, but not in the striatum. Okay? So this let me show you another example. This is actually a model that's quite widely used in our field, in the Huntington's disease field. It's a bacHD. This model, you can see what they allow us to do. Huntington gene is huge. Okay, the gene is, the protein is 350 kilodalton, 3,000 some amino acid. The gene locus is 170 kb. Okay, so that's why it's very difficult to study full-on Huntington in any system, in vivo particularly. But so what we have is now a 240 kb human back, containing all the 67 exons, and we introduce 97 glutamine repeat in exon 1, and also we strategically position these LoxP sites. So now we have a conditional back model of Huntington's disease. As a matter of fact, my lab took the same back and did a lot more further dissection of the Huntington's cis elements in disease, and it's very reproducible to sort of see some of the phenotype. Yeah.
So why did you do it this way? Could have done it the other way, where you stop it or have it flipped.
Yeah.
So
in vivo
so
experiment.
Target to one cell type instead of excluding cell types. I mean, they're two ways. They're complementary. I was just wondering what was. The logic.
So in vivo experiments takes time. Okay, and in vivo models for neurodegenerative disorders that take extra time, because you really have to wait the animal to get old to get disease. Okay, so the idea here is that we want to build the model first, and if they unveil some really interesting disease-like phenotypes, behavior as well as pathology, then we're going to do dissection, right? So if you want to switch on like baseline just in one cell type, nothing happens. Then you have to switch on everything again. And to us, the first thing to do for neurodegenerative disorder model, or actually for this matter, any brain disease models, is to do the inactivation. Of course, now we're doing the other way around. Okay, once we know it works, and because this study is all done. Okay, so once we know it works, then we can go to the flip side and try to rebuild up the disease cell type at time, or multiple cell types at time. So just quickly illustrate, we have the back. This phenotype is well characterized. Okay, it has a progressive behavior, motor, sort of a cognitive psychiatric behavior, and end up with a selective forebrain atrophy, but there's no big cell loss. What we can do then is take back HD and cross with a combination of Cree, either cortex restricted, highly striatal selective, the RGS9 Cree, or we can put in double Cree and ask, is mutant Huntington working cell autonomous in the cortex, in the striatum, or maybe there's some kind of synergy to cause the disease. We've done the study, and I can tell you is that the key finding is that we actually see a pretty significant role of mutant Huntington in the cortical neurons in many behavior aspects of the disease, and in eliciting some of the non-cell autonomous phenotypes in the striatum. And also we show that the cortico-striatal together is responsible for the majority of the behavior phenotype and the pathological phenotype we see in the bacHD model. So we show this sort of synergy and define these two critical cell types for the disease. So just to show you in the bacHD, just like in the synuclein case, we can actually cross with RGS9 Cree or cross with double Cree. We see this nice reduction of the Huntington RNA, human Huntington RNA. So the reason I'm showing you this is really to tell you this is sort of it's reproducible, it's consistent, and you should really think about adding conditionality to your back transgene so that give you one more control on where and how your transgene is expressed. And maybe if you build one model, there's many other questions you could address. Okay, so I'm going to move on to talk about the sort of the main part of my talk, which is look at using genetics to interrogate the striatal direct pathway neuronal subpopulations in opiate drug use. I don't know if the direct indirect pathway has come up yet in the course.
Only about 20 times, I think.
Only 20 times, okay, so I probably should shorten, but maybe each person has their own version of it. Anyway, this is our favorite topic, as many of you know. It's been a favorite topic, at least for myself, for many years. So the reason why we're all interested in this, at least this is my take, okay? Each of you can give me your take. So the things that, so these, I position here is the Cortico-Striatal-Thalamo cortical loop. It's the circuitry that mediates a lot of the, volitional behavior, a lot of the sort of learned behavior, and including some of the higher cognitive aspect and language. It's just the, major sort of behavior circuits for the remaining, for the vertebrates, for that matter. So people say, why don't you dissect the other part of the, the circuit? They're probably equally interesting and, more interesting. The thing is that they're very complex, okay? All these regions has many different cell types. And even with the GENSAT genetic access for some of these regions like thalamus, we still sort of not quite have good handle of all the cell types there yet. So the nice thing about striatum is that 95% neurons in the striatum all look like that. Okay, they're all medium and spiny. So if you just look for medium spiny neuron, striatum is almost like a pure cell culture. And interestingly, I nicely colored here for you. They come in two flavors. So the direct indirect pathway. So the so from that aspect, it's quite attractive from molecular and the cellular dissection aspect, right? So molecularly, it's relatively simple. We're thinking about two major projected neuron subtype. Of course, keeping in mind there may be further complexity within each projected neuron subtype, some of which we'll talk about today. So you're all famous. You all know this famous model, originally sort of described by Albin, Young, and Penny, and also DeLong. I won't go into detail except to say that sort of the direct indirect pathway, the projection eventually converge at the striatal output, and it really seems to give you the opposing sort of influence on the basal ganglia output. Many of these predictions actually pan out in some of the latest optogenetic study, especially those from Chrysler lab as well as from Eric Nessler lab. When I actually started thinking about direct indirect pathway many years ago, there was still actually some debate on how segregated these pathways are, especially at the molecular level. There's you probably heard about the debate on is D1, D2 really segregated? The reasons that people have very few markers, and you can only see these cells after you fix them. Okay, so that there's segregation, or there's some molecular signaling differences. However, physiologists or biochemists, they always see some kind of colocalization. So that is again is in a mosaic kind of distribution situation. It's very difficult to study individual cell type unless you can actually visualize them and record and manipulate them sort of separately. So the field really get a big boost from these GENSAT mice. So these are some of the original back mice that NAC made for the project. And this is D1 GIP, obviously expressed very nicely to the direct pathway targets. And this is the D2 GIP expressed only to the globus pallidus, but not the other targets. Now there's a whole bunch of other back GIP and back Cree mice that allow you to visualize these neurons, look at them when they're still alive, to do gene expression studies, and also do manipulation via the Cree. So you can do like a viral delivery, etc. So hold on, okay. So relevant to the class is that we now know these two. I mean, of course, the D1 D neuron be implicated in many sort of disease conditions. I should mention. Well, some of the reasons that we are originally interested in this is because of the movement disorders such as Huntington disease, which is being implicating the early chorea-like deficit seem to be affecting the D2 neurons. And then disease, and the in late stage disease Huntington, there's more kind of a dystonia, rigidity, and people think the D1 neuron degenerating. For Parkinson's disease, there also seems to be an imbalance of the D1, D2 neuron. So we're already interested in the pathway because of those movement disorder disease that were studied. But also, we all know that they're implicating a lot of the sort of psychiatric disease condition and addiction. So right now, there's really good evidence in the field. I'm sure you'll hear a lot from Eric this afternoon about the optogenetic manipulation of D1, D2 neurons seem to really hugely influence natural as well as drug rewards. For example, American Lobo, actually my former student in Eric's lab, showed the optogenetic stimulation of D1 neuron in nucleocompetency enhanced CPP, cocaine CPP, while stimulate D2 neuron prevent cocaine CPP. Anatol Kreiser's lab also showed that the stimulating D1 neuron in the dorsal striatum increases behavioral reinforcement. In this case, it's actually self-administration of light by the mice. And stimulate D2 neuron induced transient punishment. And there's another study also showed this sort of opposing effect of a D1, D2 neuron stimulation. So this is really just a highlight that if you manipulate the activities of those neurons, they give you this sort of opposing but really interesting readout relative to reward and reinforcement, including drug reward. Okay? So the important thing I want you to ponder is what about the genes endogenously expressed in those cells? What about your gene, or what about your gene that's broadly expressed but also expressed in those neurons? Do they play a role, in sort of a function of those neurons that are relevant to the phenotype you're interested? So I just tell a little bit what we actually did in the field. So one of the reasons that we originally want to study these neurons really to try to identify genes that are differentially expressed in this neuron. Okay, so as I told you, if you look at let's just say 1999, 2000, when we start thinking about this problem, there are not too many genes that are differentially expressed, and there's still some debate back then, as I mentioned. But with the sort of advent of GENSAT back mice, so our laboratory did a FACS sorting. Okay, we kind of used the GENSAT mice, isolate them while they're still alive. We did this at P20 and 2 months, and we actually labeled the live neurons, sort them out, and did a array analysis. Actually, recently there's another group did a FACS array with different set of way to label D1, D2 neuron. And Nat's lab came up with this really elegant method together with Paul Greengard's group, really using genetically labeled ribosome to do bacTRAP. And so you're doing, Jessica's doing some of that. So that's really nice because you sort of can do it with a grounded tissue, and you can look at the potentially transcripts in the dendrites. Keeping in mind that both methods work, and also they're looking at a little bit different kind of population of transcripts, right? So the sorting, you get a whole cell. Okay, so you get a transcript that's probably on the ribosome, but also ones that are in transit or ones that are non-coding. The bacTRAP is really nice because they look at the actively translated population. And the other thing is that I say that both type of methods work is that there's actually a nice group of genes, the D1, D2 selective genes, reproducibly observed by these studies. Okay, on the top are the known genes prior to this kind of study. In the bottoms are the new genes that's being identified. Our lab is actually being systematically sort of knocking out some of these genes to look at what their effect on development and on behavior. I'll just quickly mention one gene that we described, actually Mary Kay Lobo described in 2007. It's called GPR6. It's really interesting because I think it's probably one of the most selective sort of D1, D2 cell gene that we've found. It's also actually showed up in Nat's bacTRAP. If you look at Allen-Bring atlas, GPR6 is highly restricted to the striatum. And if you look at the GNF transcript study in the human, this is all the tissues, and that big bar is caudate putamen. Okay, so even in human is quite selective. And then we showed using this knocking, GPR6 knocking staining with a nuclear like Z, and this is D1 GIP. You see this really nice segregation in this sort of salt and pepper fashion. Interestingly, GPR6 knockout, when we originally got it, there's no gross behavior deficit. Any of the locomotor deficit we assayed are completely normal. They're pretty happy-looking mice. So we said it has to have a phenotype. It's so important. I should mention that GPR6 is a constitutively GS coupled receptor. So it's hugely pumping CMP into the D2 neurons. And so basically we finally team up with Bernard Bailey, who's a master instrumental conditioning neurobehavior scientist. Was at UCLA, now at the University of Sydney. And so Bernard basically put these GPR6 knockout mice into this instrumental conditioning paradigm. The mice learn to pressing bar for, different kind of rewards. And what's really interesting is that I will just show you one piece of data. So these mice actually acquire the reward, the task faster. When they perform at higher level. When we actually assay for motivation for reward using this progressive ratio. So every time they get one candy, they have to press 5 more times. It's a PR5. And you're looking for break point. Okay, you're looking for mice when they press, you just do 15 times 5, 75 times for a candy. They're like, that's it, I'm not doing this anymore, too much work. But look what happened to GPR6 knockout mice. They just keep going. Okay, some of these mice can go almost to the end, like 150 times, still getting candy. So they really have this huge change in their inhibition of getting reward. We now do a lot more behavior studies on these mice, and the data so far are consistent with this sort of dysregulation of this, incentive or reward-driven behavior.
I have a question about
Yes.
Mice. Did you do any like fasting or anything to get a sense if one of the two pathways is electrically up or down regulated?
Yeah, we are doing some of that, but we haven't. What we haven't done is we haven't done a lot of electrophysiology. Yeah, this is very complex behavior, so it's not really suitable for doing the kind of immediate urging study. But we have some other behavior now that are more potent and rapid. So we're trying to look at the differential signaling. Yeah, but what we really need to do, I think, is do more kind of electrophysiology. We did a little bit in dorsal striatum. We didn't see much. But what we should do is more in the ventral, in the nucleotome base, I think. Okay.
But William, this is a new target, which hasn't been really.
It is. It is a new target because it's a constitutively active and it's in a D2 cell. It has to somehow signaling maybe to modulate the way the GPCR signaling in the D2 neuron. We sort of hypothesize this since it's constitutively GS coupled, so it's likely to perhaps setting up a high GS signaling tone, so that other receptor can work against that. So why D2 neuron need a high signal tone, we do not know. So, but I think this is endogenous expressed, highly selective, and is ideal for targeting to kind of modulate this type of behavior.
Have you looked at drug reward at all, or just?
I afraid someone will ask me that question. I know it's the taped session, but yeah, we did some. Yeah, I think one thing that we pretty confident to say that is that the cocaine sensitization seemed to be enhanced. Not cocaine the cocaine sensitization, because we did the 7-day administration. During the 7 day, we don't see big difference, but when you assay at day 14, there's a significant increase. So that's the only piece of evidence I would say is consistent with the food reward side, but we obviously need to do a lot more. And I think the assay that we particularly need to do is self-administration. So I think that would be kind of parallel to what we did with the food. And maybe we can encourage Chris's group to work with us on that. Yeah.
Did you try to look at the gene of the Parkinson background to see whether you would have an incidence?
Great question. I know. Great question. I don't think we did enough. Maybe we can talk over the bar. Yeah, I don't think we did enough. But that's one of the first things we thought. Yeah, but I
They changed the progression of disease.
Oh, I see. You mean genetic model Parkinson, not the sort of a 6-OHD. No, well, the thing with the model that we're using, they're really mild, the phenotype. The phenotype are mild and they unravel over 2 years. So it's kind of a challenge.
But you may worsen phenotype or
I know.
accelerate. I don't know what would be the hypothesis behind.
Yeah. No, those are good suggestions. There's many crosses to be made. Yes.
I know it's a constitutive GPR, but have you looked for a ligand?
Yeah, I cut all the slides, try to save time. But anyway, so if you look at the structure of the
You're
protein,
interested in something new.
I know.
This is a new receptor
Oh,
for,
okay.
for
Yeah,
addiction-related disease.
So the structure-wise, the receptor belong to a family. There are 3 proteins in the subfamily GPR 3, 6, and 12. So divided by 3. But then it's also in the larger group. It's actually related to family of sphingosine 1-phosphate receptors. So there are some evidence in cell culture that S1P can actually modulate CMP signaling, but also can stimulate calcium signaling. We have some evidence, I think is not as strong, that you add, so what I can tell you is we published in the paper that if you purify the membrane, striatal membrane from wild-type knockout, actually Chris did the experiment, Chris lab, there's a big reduction in the AC activity in the whole striatal membrane prep. So this receptor is responsible for making a lot of CMPs in the striatum. Think about this whole striatum. This D2 neuron is only half of it. I think in the Nat and Greengard's paper, I think Surmire group add S1P into the wild type, slice and show D2 neuron there's a slow calcium rise. So I think the S1, but there's some negative study from cell culture, from culture cells. People claim that it cannot get the effect. So it could be context dependent. It could be that you have to have this receptor in the right cell type with the right set of signaling molecule, you can see an effect. So I think that all need to be worked out. You know what I'm saying? So what we can tell you is that this is really specific and it's important. But I think a lot of study need to be done.
Is this the same receptor that's agonized by AM251? There's a I think it's a GPR.
No, there's no other ligand that I'm aware of, unless it's unpublished.
No, it's published. I think.
AM251 is a CB1.
Yeah, it's a CB1 antagonist that actually agonizes one of these orphan receptors. And it's a GPR or something. It's the guy in Indiana.
You should send me the paper because I'm not aware.
55, is that what it is? Okay.
Okay. Question?
I was just thinking in terms of the D1, D2 sort of go, no-go storage, because this is GS and D1, D2 is GI. And I'm just thinking if you had a GS stimulant, like it should be going against the D1.
D2 you mean?
Sorry, the D2.
Yeah.
I thought you'd have the opposite phenotype.
No, this direction is right.
Like
Because if D2 neuron is suppressing, okay, right, and then this is basically setting up a high tone, so the D2 neuron has a high tone of suppression, and when the D2 receptor come in or relieve that tone. So I can tell you there's some intriguing data because this is in mice, and there's some intriguing data. I don't know how solid from kind of genetic point of view is. You guys should look up. There's a paper by Michael Frank, who's a very, very good human neurobehavioral behavioral psychologist, and he basically took some of these D1, D2 genes, famous ones as well as some of the ones that found by the field, and look at the variants and look at the behavior in human. And what's really interesting is that among all the genes they tested, they found one variant in GPR6 that's significantly correlated with human instrumental conditioning. So that's interesting. You have to evaluate it. But I think it's an interesting kind of parallel in this kind of a very complex behavior paradigm. Okay, I'm going to move on if I still have time. Oh, I really need to move on. So you say, okay, what about mu, right? We've been talking about the question of mu receptor expressed everywhere, and what I've been trying to tell you so far is that the striatal direct-indirect pathway seems to be a good place to start to actually study some of these much kind of older and clinically important questions. Because so far I showed you that the D1, D2 neuron really have good evidence involving this reward circuitry. Okay, and these are genetically accessible, and they can be manipulated in vivo. So we actually became interested in the mu in this D1, D2 neuron system from a staining like this. Okay, so mu actually not only they expressed the opioid receptor expressed in the striatum. This is known in 1976. I mean, Schneider group using ligand binding. They actually express in this patchy structure. Okay, and it's called the striosome compartment. So basically, besides the D1, D2, the striatum has a whole other layer of organization on top of it. So there's striosome, which are these patches. They actually are these three-dimensional kind of column or maze throughout the front to the back of the striatum. And the surrounding of the striosome they call the matrix. Okay. So they are originally defined actually or described by Angribio by staining, I think, choline esterase. There are many other molecular markers for either striosome or matrix. You know this, at least we think this is really important because, the original study by Anne already showed it's evolutionarily conserved. Okay, this is from cat to monkey to human. And you actually can throw mouse and rat right in the middle of this. They all have a very conserved ratio in terms of area that are either in the striosome matrix. So the matrix is 85% of the area by and large, 80% maybe. Striosome about 15, 20%. Okay. The other thing I won't have a slide for you is to show you that, developmentally, there's a clear sort of time points when the striosome neurons are born. They're the first one that's born. They're the firstborn, and then the matrix are the ones that.
I didn't think you had.
Oh, sorry, I didn't. I now have two. The striosome is the one firstborn, and the matrix is the one that's born later. Okay, so there is a sort of a precise timing when these neurons born. As a matter of fact, all the medium spiny neuron markers are differentiated first in the striosome, especially the D1 type of neuronal markers. And both the dopamine and the cortical inputs form synapse first in the matrix, in the striosome. So you can see this sort of developmental timing may be related to the circuitry assembly, but also it's hinted at these structures are important. Another reason they're important is from the connectivity mapping. Originally, I think some of that done by Gerfen's group as well as Angribio's group. So the main feature here is the following. The striosome seem to receive input, cortical input, mostly from the limbic aspect of the cortex. Okay, so we're talking about orbitofrontal, anterior cingulate, and insular cortices. So the kind of place where also nuclear compounds receive inputs. Okay, in terms of output, I'll show you evidence they're both direct and indirect pathway in the striosome and the matrix. So on the other side, the matrix is more somatosensory, motor, and association cortex. Okay, more kind of the motor sensory aspect. In terms of efferents, both matrix and striosome project to globus pallidus, the external, the indirect pathway, but also to GPI and SNR. Sorry, that's a typo here. It's a direct pathway. And striosome specific, but the important thing here, there's a striosome specific pathway. So the striosome specific pathway appear to project to the SNC where the dopamine neuron cell bodies are. Okay. So this original study, you can only kind of map approximately the processes, where the processes are. There's a study published by Uchida's group in Neuron last year. Basically, they did a retrograde monosynaptic mapping of the SNC and VTA, dopamine and GABAergic neuron connectivity throughout the brain. One of the most important conclusions, in my opinion, they found is that the dopamine neuron in the SNC and VTA has a monogenetic connectivity to the striosome compartments. So basically, the striosome neurons made a lot of monosynaptic connection to the dopamine neurons. Okay, and matrix neuron, on the other hand, is mostly to the GABAergic neurons in the SNC and VTA. Having said that, there are also some striosome neuron can also make connection to the GABA neuron too. So striosome neuron sort of can make connection to dopamine and GABA neuron in the SNC VTA, and the matrix neuron seem to be preferentially to the GABA neuron, but not dopamine neuron. So that kind of a striosome specific direct pathway idea, I think, is really nicely confirmed here. So the other aspect that the field think the striosome matrix is important is just there are many incidents of seem to be differential sort of abnormalities in various disease conditions as well as drug addiction in these two pathways, as well as psychiatric disease for that matter. So I'll give you some examples. There's a really nice review by Gio Quidden and Angribio, which I'll refer to you. So I'll give you two specific examples. One is in Huntington disease. There's a study by Richard Fall's group. Basically, he's in New Zealand, right? He's a lot of Huntington, pretty large Huntington, patient brain bank, post-mortem brain bank, and really detailed clinical information about these patients. And what he found is that their group of patients sort of, has an early motor predominant phenotype, Their group of patients has early what he called mood predominant phenotypes, more sort of psychiatric phenotype rather than motor presentation. And then there's a group of patients mixed, okay, as you would expect. So he tried to look at the two extremes and then try to correlate their behavior deficit to the pathology. So what he found is that, interestingly, the mood predominant HD patient has a sort of a big, loss in the striosome compartment. And that's not the case for the motor predominant sort of phenotype patient, both in the clinical onset and also at the end stage. So interestingly, he later did a follow-up study in the cortical area, and very interestingly, the mood predominant patients seem to have the more cell loss, both the pyramidal neuron and the interneuron loss in the limbic part of the cortex, the cingulate. And then the motor predominant patient has more kind of cell loss in the motor cortex. So it seemed to be like this sort of a circuitry corresponded cell loss in both cortex and striatum that's related to the striosome matrix circuits. The other, I think, really important study, sort of strengthening the idea, is done by Angrabios group. They basically give all different kind of stimulants to rats and look at the immediate early gene expression. All these stimulants like amphetamine, not only, so when they activate immediate early gene, by and large, are most strongest is in the striosome, which is shown here. And also, these drugs can cause stereotypy, okay, behavioral stereotypy. So they look at the degree of behavioral stereotypy and also they correlate that with the degree of immediate early gene activation in the striosome. And there's a really nice correlation. Okay, so this really suggests maybe the degree of differential activation between striosome and the matrix has a lot of this sort of behavior correlates and perhaps is relevant to pathology. Okay? So this is the kind of experiment, make people really interested in understand how exactly striosome, work and what are their connectivities and genes and etc. So, any questions? Okay. So now I'm going to try to sort of finishing up the first half by mentioning to you some of the arguments why, because we're trying to study, the point here is we're trying to study the effect of mu opiate receptor in the striosome direct pathway. So what are the evidence that mu actually is expressed in the striosome direct pathway? So there's a really nice, there's a really nice sort of a single neuron sort of labeling, projection study done by Fujiyama et al. And they basically use Simber virus to express in single neuron in the striatum, and they can label the neuron to the entirety. So you can look at all the branches in the downstream projection pathways. It's very tedious work. So they're only able to reconstruct 11 projection neurons to the entirety in the mu positive striosome compartment, and they found that 8 of them in the direct pathway and 3 of them in the indirect pathway. So very nicely, this sort of a direct pathway mu neuron they labeled. What they do is they send the little branches, they send little branches in the GPE, but then they sort of ramificate in the subsection Niagara. But they also the axonal terminal is more kind of towards where the dopamine neuron cell bodies are. If you look at the matrix direct pathway neuron, they actually is more in the GABA neuron in the ventral side. So these again is supporting maybe their direct differential connectivity, but more nicely showed in the high resolution that there are direct and indirect path neurons within the striosome, at least the mu positive area. So the challenge for mu is that Brigitte can probably give better testament to this, is that the mu in the cell, if you use antibody or ligand binding, it pretty much label diffusely the neural pill, but not the cell body. Okay, so it's hard to kind of I think some people say, why can't you just do confocal imaging to show there is this sort of in the same cell there's mu and whatever other gene you're interested. What turned out the mu is that they really diffuse. Okay, you have to actually look at the EM resolution. Virginia Pickard done tons of that, and she basically showed within the patch, 80–90% of the mu labeling processes are in the medium spiny neuron. There are features of medium spiny neuron she can look at, but it's you cannot easily do with the immuno. I would say. However, I looked into the literature. There are actually a study done before by Delft et al. Exactly Marie-Françoise Chesler's group. Basically, they did a single cell resolution labeling with a mu, radioactive in situ, but they also did ligand binding to show in the serial section. They show the patches are the places where the in situ is, and they counterstain the section with H&E. And their conclusions at single cell resolution, almost all neurons in the striosome express mu opioid receptor mRNA. So I think it's most of the neurons in the striosome express mu, and they're direct indirect pathway. Obviously, this is actually data from our lab by Yijun Cui. Look at D1 and D2 GFP and double stain with mu. Obviously, we know these D1, D2 GFP don't have those holes in the patch, so they are expressed in both matrix and striosome, and it's confirmed here. Okay, so if you put these two together, I think it's reasonable to say that D1 and D2 neurons in the striosome, they should mostly express mu. I think this argument, yeah?
Just didn't have time to show you that we have the knock-in mu cherry mice now, and we can see at single cell level where the mu receptors are. But I'll show you later.
Sounds great. I can find another study that's sort of supporting this idea. There's a really a nice set of paper published by Tony Drago's group. Okay, so he does knocking in the D1 neuron, D1 gene actually. So he made a really funky mice with a D1 locus, has a LoxP stop LoxP, and then diphtheria toxin. Okay, and then what he did is cross that with CAMK2-Cre, so there's a postnatal ablation of only the D1 expressing neurons. Okay, so he basically had in a series of papers, one of which we cite here, he basically showed there's a really nice postnatal ablation of D1 cells. You look at all the D1 cell marker genes, including D1, dynorphin SP is hugely down. Okay, but you look at D2 markers like dopamine D2 in Kaplan, it's actually significantly up. So there's a really kind of selective ablation of D1, but nicely keeping the enriched the D2 neuron. And what happened to mu? So oops, what happened? Sorry.
It vanished.
I know, that's the, this is killing me. That's weird.
You saw it for a second, and then it...
Hold on, let me just do one. I know.
Your computer's eating things, I think.
I know, it has its own mind.
Oh,
Okay, there we go. It has to be manually shown. So this is actually, recently they published studies show that if you look at the wild type mice, has these 9 mu patches from rostral to caudal, and in these D1 cell ablation mice, there's a significant reduction of mu staining throughout the striatum. So I think all these really pointed that yes, they're, the direct indirect pathway in the striatum, in the striosome, and yes, the substantial portion of them actually are the D1 neurons that are expressing mu in the striosome. So that's sort of my concluding my first part of talk, and the key question for us that we'll hope to address in the second half is that is mu in these subpopulation of neurons important for any of the opiate-driven behaviors in vivo? Okay.